So thank you very much for the invitation. I'm really sorry for being canceling last time. I was with a completely phonics. I didn't have any voice. So it would have been a total disaster. So, and I'm happy to do this today. So today, thank you also for the introduction. I will try to tell you a bit what we're working on, how we can apply tools from dynamical systems and complexity science and information theory to a very clinical question, to a very hot clinical question, which is whether people or patients that have a lesion on their brains are conscious or not, whether people who are essentially unable to communicate with us are in fact conscious of the external world. And so let me show you where I work. This is like the ICM Institute. Now it's rebranded as Paris Brain Institute, which is inside the Pitié-Salpêtrière, which is like this building over here. Actually, it's a huge hospital, one of the largest hospitals in Europe. It has 400 years. The chapel has 450 years. Luckily, our building is a bit more modern. So let me tell you a bit give you a bit of context. Right, when people study consciousness, most of them work on a question of conscious content, which means is try to understand what's occurring on a brain when we're conscious of something in particular. For instance, here you see a train, and you might see the train going in the station or out the station. So the direction which might change for you according to the way you focus on this, on the way you sort of like pay attention. And you cannot either perceive both simultaneously, so you perceive it like going in or going out. And then there's the thing that here that the stimulus doesn't change. The stimulus is always the same. What changes is the way you process these stimuli. And so there's something different on the content, and according to the brain activity, you perceive it the one way or the other. So this is just one example. There are other examples like illusion or subliminal images or clinical conditions like neglect, in which a subject can somehow like give a forced choice, correct four choices onto a stimulus, but still would say, I haven't seen anything, but I know it's there. But he says, but I don't see it. That's a clinical condition. So in those situations, we study conscious contents, and we rely on the verbal report. So we need for you to tell me, like, I'm seeing the train going to the right or to the left, and then I can compare your brain activity in one situation versus the other. And the way we contrast the activities, we actually maintain the stimulus the same, and we contrast when the subject says, I saw it, and when the subject says, I haven't seen it, when it says left versus when it says right. The question that I focus on, it's a bit like goes a little bit below this one. It's about state of consciousness. So the question is whether a system, person, animal, or non-biological entity can be conscious of the external world. And what I mean what I mean about conscious, well, here the question is whether he can report this sequence of processes and convince you that he's conscious about it. So in this case, what we're trying to assess is responsiveness. So essentially, whether the subject says, yes, I'm conscious of the external world, yes, I'm aware of the external world, etc. And the way we study this is to contrast different conditions in which the system is more or less the same, but there's a transition between reporting a conscious experience versus not reporting a conscious experience. Like for instance, in sleep, we can simplify the process on a gradient from being completely aware or conscious to completely unconscious when we are on deep sleep. Somewhere in the middle, there are dreams, so you have some sort of like conscious experience and decoupled from the external world. At some place, you lose the capacity to have a sequence of conscious events. Another example can be anesthesia. For instance, you probably had the experience of being anesthetized you're completely aware of everything, and then suddenly, the world disappears, and then you reappear into the world and you have no recall what happened in the middle. And then finally, there's the example of brain disorders or trauma. These we call disorders of consciousness. And in this case, the patients are having had an insult to the brain. That could be either a traumatic insult, a traumatic injury, or an anoxic injury. And then these patients, they tend to go to a coma, and coma means that they're completely unresponsive to the external world. And some of them wake up from the coma and they are conscious with certain level of lesions, but some of them wake up into a middle situation, which we call disorders of consciousness. And in this situation, the subjects are awake, which means that they have their eyes open, but yet they don't have any behavior that we could use to say that they're conscious. So they don't respond to command, they don't follow any of the stimuli, visual stimuli that we could give them, or auditory stimuli. So they don't orient to auditory stimuli. There's no evidence of them being conscious or not. And this is also a gradient in which we have like vegetative state patients, which we think they are not conscious. Minimally conscious patients, these are patients that have some sort of evidence of them being aware. And then there are other conditions that are along the gradient. So here the question is whether we can use tools to establish a diagnosis, to establish a criterion to separate between minimally conscious state patients and vegetative state patients that relies on the neuronal activity of these subjects. Right. So, and this question is both fundamental and applied. Fundamental because by understanding what are the neural mechanisms, what are the neural activities that might be interesting, we can understand what and how and where consciousness occurs in the brain, but also it can also give us tools that we can play on the clinic, so we can now go to this given patient and provide evidence to the clinicians to say, this patient is conscious or not, he will recover or not, and things like this. And this is a very difficult diagnosis nowadays, because even if you trust the behavior alone, people without proper medical training, I mean neurologists, they don't see these kind of patients every day. They might get even to 43% misdiagnosis. That means that they would label patients being in a coma or vegetative, while there are clear signs that they are not vegetative, and that's on behavior alone. And it's amazing, because this paper has 10 years already, and we still see this every day. We saw a patient this week, which we were called to assess him, and the original diagnosis was coma, and in fact he was conscious, but yet you need to really look into the behavior, and the behavior is very difficult to detect. So what happens if you're truly sure that the subject is vegetative in terms of behavior? Well, even that is not sufficient. So also 10 years ago, Adrian Owen, back then in Cambridge, now he's in Canada, he showed that you can ask the subjects, the patients, in question, you can tell them, now imagine yourself playing tennis, or imagine yourself navigating your house, and that elicits a very different type of activations on the brains on healthy participants. As you see here, like if you imagine yourself navigating your house, there is an activation in a region of the brain, and it sort of follows the command of saying of house imaging. And if you ask, imagine yourself playing tennis, you have an activation that now sort of follows the task of tennis. And that's for healthy participants. But what he did, like Adrian did, is to do the same thing with patients. And these patients were truly like vegetative on the behavior. They were really, really difficult there were no signs of these patients being conscious. And he found the same type of activations on their brains. So basically, on these patients, you have no evidence of them being conscious from behavior, but yet they can activate their brains following the commands and following the commands that we give, and that's sort of a clear evidence of them being conscious. And this is around 15% of the patients that are classified as vegetative. they can be on this condition. So overall, what I'm trying to say with all this evidence is that behavior is not sufficient, that the training of the clinicians alone by the clinical assessment is not sufficient. We need additional tools to assess these patients, and we need to provide them as quick as possible, because like important decisions are made every day in the acute stage on these patients. And last year we published a case on a COVID patient that he didn't wake up after the respirator was removed, and they were about to stop the care of this patient, and with our tools we are able to predict that this patient was in fact conscious, and that we still need a bit more of time on the ventilator, and he actually woke up afterwards. So this is something that it still happens every day. So what we're trying to propose is to go into a personalized, multimodal, and hierarchical diagnostic and prognostic tool. So the idea here is try to sort of propose different methods that are, you start on the base, these methods are easy to deploy everywhere, and you have a certain level of evidence here of the subject being conscious or not. And then if you need more evidence, you go up this pyramid to reduce the uncertainty of your diagnosis, but also by the cost of the complexity of implementation. And this is exactly what we do on the hospital in our team. This is Lionel Nakash, the other co-director of the team, and postdocs and PhD students, where we do several assessments of these patients. I'll go over the different. I know that it's a different audience, so I need to go over the details. So these are evoked-related potentials, so the activity that elicit on the brain to a different stimulus. This is transcranial current direct stimulation, so we stimulate the brain with electric currents to modify the excitability of the cortex, and see if we can elicit changes on the state of consciousness or in the responses on the external stimuli. This is positron emission tomography, so that we do also to assess the metabolic levels of that brain. We do EEG analysis, so electroencephalographic, we extract features, and I will go over that, and that's the complexity part that might be interesting for you, that we extract some complexity measures from the EEG and from the fMRI, the functional magnetic resonance, and we use all of that to do a full assessment of the patient. So we try to combine all this multimodal information to give a clearer idea or reduce the uncertainty related to this patient. If you want to interrupt, just don't hesitate. We can interrupt at any point, and there's something that's not clear, I'm happy to clarify. Right. So let's how do you derive markers for consciousness from the brain activity? So what we try to do something like 10 years ago already, is to go over the different theories of consciousness, and these days each of these theories has a book behind that I would recommend for you to see if you're interested on them. Essentially, it's the Parisian school from Stanislas Dehaene, the Dutch school from Victor Lam in Amsterdam. There's also the Giulio Tononi in the US, and Nicolas Schiff, which has an angle from more like clinical related. So all these different theories have proposals regarding the way information should be treated on the brain, and what is the difference on the way information is treated between unconscious processes and conscious processes. So in general, we could somehow identify common predictions among these different theories, and the common predictions are that first, you need to have sharing of information across brain regions for a subject to be conscious. So the information process doesn't have to be local on a certain part of the brain. It has to be shared, and without spatial temporal complexity. What I mean is not shared simultaneously throughout the brain, but it should have like follow a certain level of complexity that goes from one location on the brain, then to another, then back to another place, and so on and so forth. Second common prediction is that the information has to have a level of complexity, and what I mean by that is that the level of complexity of the external world, the level of information that is in external world, it's determined. We have a level of information to be processed, and if we somehow like map that into brain activity, if we do a representation of that into the brain activity, the level of complexity, the level of information of that data should be sustained. Otherwise, there's no good representation. You have a more complex environment, you should have a more complex representation of that environment. So that's also an important prediction from this series. Third point is that when we're processing something, it has to maintain in time somehow. Like it doesn't have to be brief. Doesn't have to be too fast or too slow. So there's sort of like a sweet spot in terms of the information be sustained in time in order to be stored afterwards. And then in terms of like interaction between regions and more like a model of neuronal masses, certain frequency bands or like oscillations should be more relevant than others. So essentially what we tried to do back then is to quantify these different predictions into biomarkers and test how good they are to characterize these patients. And the second thing that we tried is to try to combine this information from different predictions and see if we can create a better classifier for the state of consciousness of patient by combining these different biomarkers. So the first prediction was like this information maintenance and evoke rated potential. So for that, we develop a protocol of stimulation, which essentially has two levels of regularity. So it's an auditory stimuli. With these patients, it's hard to do visual stimuli because they don't focus their attention on visually, but the auditory attention doesn't have a spatial resolution. So what we have is like two levels of regularity. There's a series of tones that can either be all the same or have the last tone different, as the figure shows. And that can either be like frequent or infrequent. What I mean by that is that here in this case, like what happens sorry, let's complete the figure. So here you have like these two types of stimuli that they can either be like frequent. What I mean is like you can have this thing to happen like very frequently, or rare. Like in this case, like in this sequence of tones, this overall sequence is more rare. It happens only 20% of the time compared to all the ones here. So basically, what we use this for is to separate two types of responses that occur on the brain. The first type of response is the detection of change. So our brain is very good to detect changes, and that it reflects into evoke potential, which is called mismatch negativity. It's basically a negativity that occurs around 150 milliseconds after the stimulus onset. And that detection of change on the brain, it's very fast, and it's very it's sort of like resistant to all kinds of manipulation. So you can have this detection of change when you're asleep, when you're anesthetized, when you're vegetative. So it occurs like almost independently of the state of consciousness of the patients. On the other hand, the detections of regularities, long-term regularities, to detect there is something that is frequent or not, it's related to things that occur later in time, so around 300 milliseconds after the stimuli, and that only happens when you are aware and attentive of the stimuli. So basically, we separate this, what we call mismatch negativity, a negativity around 150 milliseconds, and P300, a positivity around 300 milliseconds after the stimuli. And these two types of features that we can extract from the brain responses are very good to detect consciousness, because this one disappears when the subject is not conscious. So, what you see here is in the type of responses that you see on a healthy participant, the type of responses that you see on a vegetative patient that doesn't have is not conscious, and the type of responses that you see on a patient who's vegetative, but in fact was in a locked-in syndrome. So it was completely conscious, but it couldn't behave in any way. So you have these type of responses that we are describing. So, but that's sort of very limited in terms of or performance is very limited. So more recently, what we try to do is to do machine learning. So for that, and that works very well. So what you can do is you can do the analysis at the single electrode level, so one electrode on the head, or you can do the analysis using machine learning, using all the electrodes at a given time point, or you can give all the electrodes on all the time points, and using machine learning, you find an hyperplane to separate the conditions and see if this works or not to classify the patients. And that's exactly what we have. Yeah, there was a question? No. Okay, sorry. So I'll try to focus on this figure here. So what you see here, essentially, this local effect, the one that was present independently of the state of the subjects, we're able to detect it in 51% of the vegetative patients, 65% of the minimally conscious patients, and 70% of the, we call this exit MCS. So these are patients that are really lesioned, but it's clear they are conscious. That's the local effect. So it's the effect that should be happening in all cases. But in the case of the global effect, which is this thing that occurs only when you're conscious, we're able to detect 15%, 14% of patients with a global effect. And these patients, we think that are in what we call a common motor command dissociation. So we basically, there's a dissociation between the state of consciousness and the capacity to behave. So that was our first approach. So to assess the brain responses of these patients to the auditory regularities. But then, we wanted to go a bit further, so we tried to assess this information sharing across brain areas as a potential mechanism also for consciousness. So for that, I developed a new tool, which essentially what you have with EEG is time series. So time series of electrical activity in different locations of the brain. And the problem with this time series is that they are these recordings are extremely noisy. They can also be, you might have two electrodes which are basically reflecting the same source. So they are not independent. So all the traditional methods of assessing the level of information sharing between sites could not dissociate between the different types of patients. So what I did is to apply a method which is called permutation entropy. You might heard of it. It's essentially what you have is a time series. And what you do with the time series is to transform the time series into a sequence of symbols. And the symbols represent different shapes that might occur on the data. So for instance, if the data is going down, you have a sequence, an order sequence like this. You put an A, so you put a letter A. If you have a sequence like this, you put a B, a C, D, E, and F. So essentially what you have is a kernel of a certain quantity of elements, in this case 3. And when you have 3 elements, the way you can order 3 elements is 6. So it's basically the factorial of 3 is 6. If you put 4 elements, you have 24 different symbols. So the original paper published in Physics Review E on permutation entropy basically what they did was to quantify the occurrence of the different symbols and compute the entropy according to the probabilities. What we did here is when you have two of these signals, you can do compute mutual information. So just to measure the co-occurrence of these different symbols and fill a matrix like this one here with the probabilities of the co-occurrence of the different symbols. And the interesting thing here is that now we could sort of somehow quantify nonlinear couplings. So essentially, let's say in this example that I'm giving you here, each time there is an A on the top signal, there is a B on the signal below, and so on and so forth, like A to B, B to C, C to D. And if you do any kind of linear computation of information sharing between these two signals, you find it equal to 0. So so you're not seeing the relationship between these two signals. So the advantage here is that we are able to quantify nonlinear couplings. In addition to that, I told you that there is a problem with EEG, that you have like common sources and what we call volume conduction. So the electrical signal, you can see it like in two places, just because of the fact that it's an electrical medium and you have a propagation of that signal in two sites, and you have like an spurious relationship between two sites just because of the electrical medium. So what we did, and the hypothesis here is that this spurious interaction will be seen only in the diagonal because you have a zero lag relationship between these two spurious signals. So what we did is to penalize with weights equal to 0 the diagonal. So overall, the combination of permutation entropy and mutual information and penalization, we put it together into a new measure we call weighted symbolic mutual information. So weighted because of the weights we applied, symbolic mutual information because it relies on the symbols, and the mutual information. So when we apply this new method that we propose on the data coming from the patients, we saw a really nice phase transition, we could say, with phase transition between vegetative patients and minimally conscious patients. So you see a huge jump in terms of information sharing. This is a topography. So essentially, in each location, you see the amount of information that electrode shares with the rest of the brain. So it's not a bivariate relationship. The bivariate, you can see it here below. But you see a huge gap between vegetative patients and minimally conscious patients. And that overperformed any other type of measures of connectivity of information sharing. And since then, since the original publication in Current Biology, this method has been contrasted to other methods of information sharing, like WPLI and things like this. And that's an important advantage in terms of penalizing common sources, but also detecting nonlinear interactions. And one of the advantages also is it's a method that doesn't require a lot of data. There's a lot of methods to quantify coupling of nonlinear systems, but this method it's not that greedy in terms of data. So it was quite efficient, and we're using it still a lot to characterize these patients. And there was also a nice prediction in terms of like the distance between sites playing an important role to distinguish patients. So what you see here is that if you atomize the analysis for different distances, like short distances on the left and long distances on the right, you see that the differences between the different kinds of patients only appears when you look at the longer distances. And it's not because of the overall connectivity being penalized, it's because, like, when you see short distances, everybody's the same, but when you go into even longer distances, the vegetative patients go down, and the healthy participants and the conscious patients go up, right? So then, in a company paper, we put this measure along with several measures of complexity, connectivity, etc. And we try to compare the performance and see whether they carry independent information or not, and whether we can optimally combine these different measures to have a better classification of the patients. So in that paper, 2014, we computed 92 different EEG biomarkers. The first ones are these like event-rated potentials I mentioned before, some like Fourier decomposition of the data, then measures of complexity, and I might spend a few minutes here to tell you the different types of complexity that we assess. Like for instance, here this is spectral complexity, spectral entropy. So it's essentially normalizing the spectrum of the signal and computing the entropy as if the spectrum is a probabilistic distribution. The permutation entropy, which is essentially computing from the symbols the entropy of each channel, and you see that there is sort of like an increase of complexity in the different electrons when you compare vegetative patients and minimally conscious patients. And then there's another type of complexity that we compute, and that's quite interesting. It's related to what Giulio Tononi proposes. It's the Kolmogorov complexity. So essentially what we do is we code the EEG data into a sequence of symbols, and this time, instead of computing the entropy, we try to compress that sequence of symbols, with the idea, the Kolmogorovian idea, that the compression, it's a nice approximation of the algorithmic complexity. What it means is that the shortest algorithm that can generate that sequence. And what you see is that effectively, like in the vegetative patients, like the overall signal can be easily or more easy compressed than in healthy participants or in conscious patients. So essentially, the overall sequence of symbols, it's harder to code in conscious subjects compared to vegetative subjects. And then like, there's the WSMI that I mentioned before. So overall, in the original paper, there was like 92 different features, and you can see the topographies of the different features in the different patient groups. Some of them work better than others, like the permutation entropy works very well, WSMI works very well, different spectral bands works very well. We can easily like reduce that information into a single value and then compare whether they are good or bad. Like this is just like the red balls are the vegetative versus minimally conscious comparison. If you're farther from the 0.5, that feature is good or bad. So you might see that some of the spectral measures are good. The information theory and complexity ones are also good. And also the connectivity ones are good. And then you can average the signal across all trials, or you can measure the fluctuation of the signal across trials. What I mean is like the variance that you have during the recording, and that can be informative as well. So overall, you have 92 features, and then you can like normalize it, reduce it in different ways. So it ends up being like 400 different features per subject. So, now we have 400 features for subject. Is it like better than have only one or the best one? And the answer is yes. So what you have here on the left is the performance of the best feature. And this is classification, around 70% classification performance. And if you do like I'm sorry, you don't see the labels, but if you now compute the performance combining all these features, you gain around 10% of performance. And that's very important, because 10% more, we see around 50 patients per year or more. That means like 10 or 5 or 7 more patients that leave the hospital with the right diagnosis. And then like, so this is like the group level, what happens at the individual level. So at the individual level, we can try to compare the diagnosis of the doctor with the diagnosis we provide with our computational system. So what we saw is that in fact, there's a 2/3 match. So when the doctor says it's vegetative, in 2/3 of the cases, the system says, yeah, that resembles a vegetative patient. But in 1/3 of the cases, it's not. There's a mismatch between the clinical diagnosis and the computational diagnosis. So, this could mean that, we are, not precise enough, but it also mean that we are seeing something that the doctor is not seeing. And to assess that, what we did is we compared the level of recovery on the two cases. And the level of recovery, when there is a matching diagnosis between the clinics and the computational prediction, it's around 1/5, 20%. And when there's a mismatch, the level of recovery, it's double as much. So it means that we are seeing something on these patients that, it's relevant. And that's why we since 2014, we are constantly using these type of methods to assess the patients. There was like a more recent paper that validated the data with an external lab. I won't go into that. But essentially now we have like all this pipeline of analysis and all the tools to compute the different biomarkers is available online. So you can there's a demo over there if you wanna check it. But essentially you can upload the data from your patient and run the analysis and get the prediction for that patient. And this method it's now used in more than 6 or 7 hospitals in the world, and we are trying to extend the model to other pathologies to predict things like deterioration on mild cognitive impairment that leads to Alzheimer and other pathologies that are also interesting. So overall, that this is we have also a company that's of course trying to be pushed with these results. So that's sort of the thing that we do with EEG. That's very related to the assessment of different measures. I had other like sections of my presentation. I would pass this one quite quick because I think we have time. Essentially, if you want to hear a gist of it, we are also trying to assess like the relationship between consciousness and the control of the body. So our brain is constantly controlling our body, the heart, respiration, etc. And that somehow like the conscious processing affects the body. And what we're trying to assess is indirect measures of consciousness from the body. And we have a nice result recently published, where we measure like synchronization of the heart when we are listening to the same story. And we show that in fact, when we are attentive and conscious of the story that we are listening, our heart synchronizes. And we can use that for assessing whether the subject was aware of the story. So let me show you only one result on that. So this is the level of synchronization when you're attentive to the story across subjects and when you're not attentive to the story on the heart. And in fact, that predicts the level of synchronization that you have. That predicts your capacity to remember the story, to remember elements of the story. So in fact, if you're really connected to the story and you pay attention, you are more synchronized to the group, and that predicts that you are able to back then, like afterwards, remember elements of the story. And we are using that to assess patients. And then I will close with a very short story, which is actually the paper that you used to invite me, which I think it's also very interesting because we try to assess some of these predictions that I mentioned at the beginning, not on EEG, but on with fMRI. fMRI, it's a method that measures the oxygen consumption of the brain. So the advantage compared to EEG is that we have spatial resolution, so we know exactly where the information is coming on the brain. The disadvantage of fMRI in comparison to EEG is that we lose information regarding temporal resolution. So the EEG has a temporal resolution of around 100 milliseconds or less, 10 milliseconds, I would say. And the fMRI has a resolution of around 2 seconds. So there are different types of like things you can observe with one method or the other. So the original result analysis was done on monkeys. We study fMRI data from monkeys either anesthetized or awake. And what we try to see is how the different areas interact to one another. So this is what happens, like the different interaction between brain areas during 20 minutes. If you average the interaction between brain areas during 20 minutes, and this is the interaction between brain areas on an awake monkey, and this is with moderate or deep sedation. So you see that, you know, even when you're sedated, even when the monkey is sedated, there are plenty of interactions still present in the brain. But you know, clearly the map is not the same. Clearly there are negative correlations between brain area that disappears. But overall, like, if you forget about the negative relationships, overall the map seems to be, you know, similar. You have long-range interactions. You have like blocks that are still present in deep sedation and awake condition. So our hypothesis here to disentangle why they were still quite similar, the interaction between brain areas in anesthesia or awake, was to say, you know, look, the problem here is that you analyze data during 20 minutes and you assume that the activity, the brain activity, stays the same during the 20 minutes. And it's clearly not the case. Clearly there should be some kind of dynamics behind the brain activity that should be relevant to consciousness. So in fact, if you decompose now this correlation between brain areas in time, you see that in fact, there's not a static photo of interaction between brain areas. So you might see, like here, I'm decomposing the activity using sliding windows, and you see that, you know, like sometimes there's an area that interacts with another, and then like cease to interact, and then it interacts with another area. And there is sort of like an exploration of different brain configurations that it's relevant. So it's not clearly not a static picture. So there is some sort of information on that data. So what we did is essentially decompose the time series into snapshots of interactions, of maps of interaction between brain areas. You do that by using a sliding window. That was on the original PNAS paper, and now we use other techniques as related to Hilbert decomposition of the signal and a coherence, instantaneous coherence between regions, and that, you know, gives you like instantaneous map of phase locking. So anyway, so the point here is to have like snapshots of brain configurations. And now you can stack all those snapshots and put them on a high dimensional space. So here it's just an example, a fake example. But you know, this hyperdimensional space, essentially each dimension is the interaction between two brain areas. So each photo, it's a point that describes this interaction between different brain areas. And once you have that data in this hyperdimensional space, you can do unsupervised classification. So you can try to sort of cluster the different configurations that are observed across all the different sessions. And once you do that, you can extract those like clusters, and you know, using k-means in this case, you can, you know, ask for as many clusters as you want. Our results didn't change with the number of clusters required. But essentially, you can see like there is a clear exploration of different brain configuration, this time clearly different brain configurations. And we order these configurations by the similarity of the map between the functional configurations, so the functional interaction between brain area, and the structural configuration. The structural configuration is the wires between brain areas. So essentially, the wires always stays the same. The wires between the brain region A and B are always the same. What changes is whether there's a functional, effective connection between these 2 points. So what we did is we sorted the different states according to the similarity. So on the left, there's the less similar configurations to the structure, and on the right, the most similar configurations to the structure. So that's essentially what I'm trying to show here. So this is the structural configuration, and on the ones on the right are more similar. Clearly they're not the same, but you just measure the correlation between this matrix linearly and this matrix linearly, and each one of them is matrix linearly. So what happens if you now quantify the occurrence of these different brain configurations according to the state of consciousness of the animals? So briefly, what you see is that when you look at the awake monkeys explore the different brain configurations more or less equally. So they explore all the configurations. So on the right, the ones that are similar to the structural connectivity. On the left, the ones that are less similar to structural connectivity. So they explore more or less all of them. So on the x-axis, the similarity score of the structure of the configurations, and on the y-axis, the probability of the different configurations. So it's a flat relationship. On the other hand, if you look at the sedated animals, you see that the states on the left completely disappear. They're actually equal to zero. We don't see them on the exploration of the anesthetized animals. So overall, what we try to say here is that the dynamics of exploration of brain states, it's completely different when the monkey is anesthetized compared to when the monkey is awake. And essentially the functional configurations, the one that do not directly rely on the wires, disappear. So last year, well, actually 2019, it's like 2020 hasn't existed. So we published the same results on patients, and this time was a multicentric study, including data from us in Paris, data from Liège in Belgium, data from Nicolas Schiff in New York, and Aaron Owen in Canada. And what we saw is the same types of configuration, this time 4, but if you do it on 7, it's the same. And what you see is that these functional configurations, that has negative correlations, it has long range interactions. It's not present on vegetative patients, and it's predominant on healthy participants. And vice versa, the one on the right, the one that really resembles the structural connectivity, it's predominant on vegetative patients, and it's less frequent, but yet happens on healthy participants. So essentially, if we compare now the results that we had on the monkeys, remember the flat curve on the awake monkeys and the steep curve for the sedated monkeys, and now we do a similar analysis for healthy participants here in green, minimally conscious patients. These are patients that in principle are conscious in blue, and vegetative patients in red. You see that there's sort of the same fingerprint in terms of the exploration of brain states between when you compare healthy participants, patients, and monkeys. So it seems like this dynamical fingerprint, it's valid across species, but also valid across ways you can lose consciousness. In one case, anesthesia, in another case, brain injuries. Also, we tested the assumption, the hypothesis, the sequence of states is going to be more predictable for vegetative patients compared to healthy participants. So not just the probability of occurrence, but overall our capacity to predict what's coming next. So once you are in one state, what's the next state that comes becomes more entropic, becomes more harder to predict with healthy participants compared to vegetative patients. And I won't go into this control because it's very clinical. But a few take-home messages, like we now have like a valid method of computing consciousness or estimating and predicting consciousness from EEG, and that's been translated to a web-based automatic pipeline, and which is now available for use for all clinical centers. I haven't talked much about the brain body markers, and this can provide new clinical tools. And I'm quite interested on these new clinical tools because essentially these tools are quite cheap, and you can apply them in any complexity center, in any hospital, independently of the level of complexity of the center. Even in the ambulance, you can do this kind of assessments. And the validation of dynamical resting state fMRI markers of conscious states, from starting with the monkeys and now with humans in the clinical setup. And I haven't talked about simulation, but we also validated the fact that when you stimulate the patients with electrical stimulation, these biomarkers actually change and track the impact of the stimulation. They can be used to tailor the way you can stimulate to enhance the performance. Let me give you a couple of words of what we're going next. So together with Gustavo Deco and others, we are now trying to go into the modeling of each individual patient. So if you can translate each individual patient from their EEG, fMRI into an in silico model, you can try now to extract biomarkers from the in silico model. And these biomarkers, we classify them in two levels. We call them first level biomarkers and second level biomarkers. The first level are basically the features of the model. So you feed the model and you end up with a level of a certain number of parameters you can use to contrast and predict each single patient. But furthermore, like once you have the model, the in silico model of a single patient, you can now stimulate the model or perturb the model, and many other ways you cannot do on empirically. So this second level it's what you do on your dynamical system. So you push it and you modify it, etc. And you can use that also to characterize the subject. And then finally, like the idea is that once you have this model of the subject, you can try to predict what's the optimal intervention for that subject. Like for instance, if you do electrical stimulation, so you essentially change the excitability of a certain region of the brain, will that bring that model and then eventually that patient closer to a dynamical activity that resembles more the activity of a conscious subject, or do a direct brain stimulation. So you put, some people do put electrodes deep into the brain and stimulate the thalamus to project and activate the brain activity as a whole in the brain. So would that work or not with that patient in particular? So you can somehow test that idea on the model before you actually do it on the patient. So that's essentially what we're trying to go. And thank you very much for your attention. I hope that you were able to follow me, and then I'm looking forward for your questions. Thank you.
Thank you very much. Really well, really very interesting material. I mean, there's a lot to cover, and of course we're not experts in this, but we get a glimpse of what's happening here. That's really nice. So yeah, people can ask questions in the chat, and I don't see anything yet. But what came to my mind, and I find it very fascinating. I mean, can you now sort of from all these analysis better define what consciousness is? I mean, it's a very, you know, very abstract, say, concept, but I mean, is it now better to define?
Yeah.
I mean, it looks like
the definition of consciousness is it's again it's a problem of it's more a philosophical problem than empirical problem to me. You know, we always try to stay on the pragmatic side of this, in the sense that we rely on the clinical definition of consciousness, and we try to provide using, you know, the methods that I mentioned, way to contrast, like, given the labels, right? You know, what we can say today, you know, is that, you know, there are questions related to what areas of the brain are really relevant for consciousness. And at the beginning of this process, like the most of the predictions were related to frontal areas, like related to control and to the representation of the process of the information.
Yeah.
These days, I would say, you know, clearly the back, it plays a more important role, at least for a state of consciousness. So the fact that, you know, we are able to process the information on the back of our heads, it makes a clear distinction, at least for the state of consciousness of the patients. To me, you know, like this, like in the field of consciousness, there's a strong debate whether consciousness it's on the front or on the back. To me, you know, it's clear that both are necessary. Otherwise, you know, our conscious experience will be completely different, because even though, like, the front of the brain might only, like, give context and social, would say, you know, context to our conscious experience. Without it, you know, our experience will be completely different. So yeah. Though I think we have provided a bit of evidence for to understand better how consciousness occurs in the brain. We also validated, like, these predictions. So, like, most of the predictions regarding interaction between brain areas and the complexity of the information, both in terms of space and time, were validated by our empirical data, the quantification of that and the empirical data.
But I mean, your experiments with the monkeys, right? I mean, it looked like that if you are awake, it looks like the interconnections are more in patterns. So at a higher hierarchical level than what you have when you're sort of unconscious, right? I mean, in the state where you basically, the patterns follow basically the neural connections.
Yeah.
So that is something.
But the thing is that, you know, that only appears when you look at the dynamics, when you look at the sequence of states that occur.
Yeah.
It seems that there are states that disappear when the subject or the animal is not conscious, but it's not 100%. So I mean, it's not that, you know, they completely disappear. They might see, like, some glimpses here and there. And one of the other experiments that we were trying to implement is do real-time analysis of the fMRI, so you can detect these, like, outliers or, like, mismatched states, so mismatch between the brain activity and the clinical state of the subject, to detect, like, potential better windows to interact with, to assess and interact with these patients.
Yeah.
So that's sort of like one side of the coin, like to say, no, let me detect when this patient has this special pattern, to be able to assess him and interact with him. But also there's an interesting point that we like healthy participants and awake monkeys spend half of our times with exploring states that are related to unconscious patients. So what does it mean? We are unconscious of the external world half of the time. Well, I don't know. It's an interesting question. I want to try to assess it. How we, like, basically filling the dots, like, in between samples of data, it's an important question, I think. I believe that our brain is essentially sampling the world in a high frequency, but we don't have like so there's an unconscious sampling of the world at high frequency, but we don't consciously sample internally that information at the same frequency. So essentially, there should be, I think there is a kind of buffer that storage data, and we essentially sample that if there's no surprises or nothing that grabs our attention, and then we integrate the points. And then if there is something that grabs our attention or creates an alarm signal, something that should really put us on focus, then it's bottom up that brings the attention or brings consciousness to it. So it's an interesting line of research that I'm trying to implement currently. I did the first. We did the first pilot last week on that.
Yeah.
Essentially that, a real-time fMRI analysis and then potentially move it to EEG, so we can put it move it to the bedside of the patient.
Yeah. Okay. Anyone? Any question of? I mean, I have one bit more technical detail. I mean, you use this ordinal analysis, right? I mean, basically these patterns to determine nonlinear sort of correlations. Is when you just do say you make an interaction network, so you just look at Pearson correlations between those time series. Are they really very noisy? I mean, don't you see any significant connections between those?
So actually, you see connections, significant relationships between sites. The problem is if you now do a statistical analysis to try to distinguish. I'm going to stop the screen. To try to distinguish between vegetative patients and minimally conscious patients using like Pearson correlation in both cases, you don't see any difference. Essentially, there's sort of like the Pearson correlation, the linear correlation between brain areas, it's dominated by spurious interactions that they don't seem to be relevant for consciousness. So there are strong relationship between areas that they don't seem to be relevant for consciousness.
Yeah. Okay. Well, I regret that we're relatively few people today. I mean, I think this is a really fantastic subject also for complexities. I mean, what is more interesting than the brain, right? In terms of what kind of behavior it can induce. And I saw in the past, I saw some movies on how this neural signaling goes. I mean, it's really fascinating how that proceeds in the brain. So, but I don't see any more questions. And so let me thank you again and all others who've been present. And okay, I thought there was a question, but it's a thank you.
Thank you for your attention.
So, much success in your further work. It really looks, really at the boundary. And thanks again for sharing it with us today.
Thank you, and good luck.
Yeah, okay. Thank you. Bye-bye.